So we're going to be we're going to be starting a protein gel, and this is 3 steps. Step 1 is going to be preparing our samples, and step 2 is going to be preparing our gel box. Step 3 is going to be loading and running our gel. So what I've done is I've taken our protein liquid that was in our 24-well block, and I've transferred 20 microliters into these 5 tubes. 1, 2, 3, 4, 5. And then I've set up a bunch of loading buffer in here. So I'm not doing this perfectly exact. I'm just kind of eyeballing it. So I added about 5 microliters of water to all these tubes, 7 microliters of 2x loading buffer, and I'm going to be adding 1 microliter of protein, 2 microliters of protein, and then I'm going to be doing a couple of custom runs here. And our protein gels have 15 wells, and I always like to fill every single well. I think that if you're going to be using some sort of a resource, you want to get all the data out of it that you can. So, I'm going to take my multi-channel, set to 1 microliter. I'm going to stick 5 tips onto it, and I'm going to go into our protein. I'm going to pull up 1 microliter, double check everything. I don't know if you can see that. Hopefully it comes out. And I'm going to pop it in to our loading buffer. Double check it. Okay, I've ejected. And then, since this is kind of waste, I can go back in there. I'm going to double dip. Normally you don't want to do that, but again, I've just aliquoted out. So next we're doing 2 microliters. So, oops, looks like I missed. Oh, no, looks like I got it all. So 2 microliters. Hopefully you can see that shadow. And that's going to go into here. Make sure that my pipettes are empty. Okay, so you can eject that tip. And you always want to write down what your samples are, which I've already done over here in my own kind of hard to understand code. So I've done 1, we're good, 2. Now I'm going to do 4 microliters of Cree and our mixtures, just because I think that we're going to have a very faint signal from that. So I'm going to pull up 4 microliters of Cree, eject it into that one, which already, again, has our loading buffer. And I am switching tips now because we're doing different samples. Then we're doing our 2 different co-precipitations. So a new tip, to do some sample, check the tip. And then 4, 1, 2, 3, 4, that's 3. And then I'm actually going to do a 6 microliter run of our Cree, because that is going to have the faintest signal if there is any signal. It's going to be the last one. And it would be good if I had a his tagged Cree so I could run it just to see what the pattern was. And I'm going to omit this for now, but we're going to come back to that a little bit later. So now that my samples are all aliquoted, I'm going to do a couple of things. First, I'm going to move this out so I can cap them. And I'm going to cap. Have a little bit of excess cap there. I'm going to chop that excess cap off of the razor blade. I should cut away from myself, I guess. For all the Boy Scouts out there. And cut off there. And I haven't rearrayed my tubes or anything yet, because I want to number them. So it's very important that you keep track of what your samples are, because if you pick, let's say I pick this up, and then I go like this, I'm like, uh-oh, which sample's which? So the first thing you always want to do is definitely label. So I'm going to use a very simple labeling system. 1, 2, 3, 4, 5. Kind of gives you directionality and what is what. I'm going to continue. 6, 7, 8, 9, 10. And then we have 11, 12, 13, 14, 15. So our loading buffer has a little bit of Comasi and a little bit of SDS. That's going to denature the protein and then cover it in Comasi and that's going to allow the protein to run. So in order to kind of complete this process, we need to have full denaturation, and that requires a little bit of heat. So we're going to come over to this thermocycler. I'm going to kind of tap them down. You know what? We should probably do a little bit better of a job mixing this, so I'm going to give a quick flick. A little quick flick. Make sure it's all nice and mixed up. Nice quick flick. Tap them down. And pop them in. Making sure that there's not any liquid on the top, because if there was, I might centrifuge it down. So our samples are now in the thermocycler. I'm going to tighten the thermocycler. Go to block. Go to run. Go to, I think it's under main. And then we have a program called 95. Tubes. Doesn't really matter what volume. Heater less. Heater lid, yes. And then this is going to run for 10 minutes at 95 degrees. After which point our proteins will be ready to be run on a protein gel. And that's it for now.